Hello everybody, my name is Dr. Ucsc. I'm an associate professor at Duquesne University, and today I'm going to show you how you can access data and download it from the Ucsc Genome Browser, get it into Galaxy, manipulate the data, and then import it into RStudio. So we're going to start off at the Ucsc Browser webpage, and we're going to select Genomes, and then we're going to go to the human genome. Now, what the Ucsc Browser is, it's an excellent way of portraying different data and information on the genome that you're looking at and superimposing it on the chromosomal region that you have in the viewer. And so basically all the piece of information or the data sets that are, or databases that are being projected on the chromosome are basically represented and visualized in tracks. So I've highlighted the gene code track for the gene annotation of the human genome assembly, okay? And so what's really cool about the browser is that all the data that you can visualize in these different tracks, you can actually download and work with and analyze in your own research or in any endeavors that you might have. And so for example, today we're going to work on accessing the genome annotation data for the human assembly, and again being able to import it into RStudio so we can do some analysis on it. And so the way you can get this data is you go to Tools, then Table Browser. And in the browser it's going to give you these different options of what kind of data that you want to actually download and access. And so we're going to select the group genes and gene predictions because again we're interested in getting all the genome annotation, or I should say the gene annotations, which is basically the location and start and stop points for genes, where the exons are, where the UTRs are, etc. And so within gene prediction group, we're going to select the NCBI RefSeq track. So again, this is going to be a list of all the genes that are in the RefSeq database and annotated in the human genome assembly. Next, we're going to make sure that we select that we want to get this data for the entire region, okay? Then we're going to go down to the output format and we're going to select all fields from selected table. And we're going to ask the table browser to send all that information to Galaxy. Next, we're going to make sure we have plain text selected. Now, before we actually send it to Galaxy, let's take a look at what is in the table, right? To make sure that it has the information you need. So to do that, we're going to go to this little tab here that says Describe Table Schema. And so this basically is going to tell you what's in the name of the column. So the field would be the name of the column, right? So we have bin, the name. So this is the name of the annotation or the transcript that is in that particular row, chromosome that it's from, what strand it's transcribed from, the transcriptional start site, the transcriptional stop site, the codon sequence start site, coding sequence start site, coding sequence end site, the exon count for the gene or transcript. So in some of the genome annotations, they're actually giving you an annotation for all the different transcripts that have been described for that particular gene. And then we have also, in addition to the exon count for the gene, we have the start sites for all of the exons. We have the end sites for all the exons. We have the score. Well, I'm not really sure what the score is. We have the name 2. So name 2 would be the official gene symbol for that particular gene, right? So if you have multiple gene transcripts, you're going to have all those designated with the same gene code. And then we also have a column that has the start positions for all of the, let's see, oh no, okay, so yeah, okay, so I think I already went over the row. So this exon starts is where each of the exons start, exon end is where each of them end. Towards the bottom, that CDS start stat is telling you what is the status of the coding sequence annotation. So either it doesn't have one, so it's not a coding gene. So for example, let's say long non-coding RNA or microRNA, it's unknown, so it's probably just computationally derived, or it's incomplete or complete. And same for the coding sequence end sites or end positions, right? So basically how much information there is to say that this is definitely the end of the coding sequence. And then the exon frame, right? So basically, one thing people don't necessarily appreciate is that each exon can actually start at a different frame position, right? So basically, some exons will start, translation will start from position 1, for some it will be position 2, for some it will be position 3, okay? All right, so this is just to give you an idea of what exactly is in this table. Now what we're going to do today is our goal is to figure out what is the mean length of all of the genes in the human genome. Now this table basically has the the transcriptional start site for each gene or transcript, and it has the transcriptional end position, right? And so basically if we want to get the gene length or the length of that transcript, we would have to subtract the transcriptional start site from the transcriptional end site, right? And so to do this, we're actually going to do that in Galaxy. There's a really easy way that you can manipulate and compute things on large tables in Galaxy. I'm going to show you how to do that. Okay, so let's go back to that table browser. We have all our fields selected, and then we're going to get the output. We're going to click Get Output. And we're going to send the query to Galaxy, all right? Now, you're going to get a little tab that say the job has been submitted, sent to Galaxy, and then the Galaxy website is going to open up. The way that the Galaxy website is organized, in the middle of the window, there's a Viewer section, okay? On the left, you have a list of all the different tools that are available in Galaxy. And on the right, you basically have a section that shows all the data sets that you've imported and potentially modified or done analysis on, right? And so it basically has this based on the history of how you've been working with it. Now, I'm not logged into Galaxy, but if you actually log into Galaxy, create a free account and log into it, it's going to basically save all those different files that you imported and modified, right? If you don't log in, then you're not going to have that next time you open up Galaxy. But I don't need it, so I'm not logging in. Now, when you import a file or a data set, initially it's going to be brown or gray. So it's going to be, I think it's gray when the import is basically scheduled, and then it turns brown when it's being processed. And then once that job finishes, so basically what you're doing is you're sending this information to the server that all the Galaxy software is running on. The server processes it and then sends it back to your web page, okay? And so once this data set turns green, that means that job finished and it's available for you to view it. And so if you click on the actual name, it's going to basically give you approximately just some basic information on the file. So this is a table that's around 110,000 lines. And then it gives you a little viewer that shows you the first 5 or 6 lines, so you have an idea what's actually in there. So notice again that all the columns have a name, which basically comes from that field variable that was in that table description that we saw in the Ucsc browser, okay? Now, if we actually want to view that table in more detail, then all we have to do is click this little eyeball, View Data, and now that table shows up in the middle, right? See? It's all the way across, right? So there's 16 columns altogether. Now, first thing we're going to do is we don't need all these columns, right? And so a lot of times you're going to have these large files where you're going to potentially have additional information that you really do not need, right? And so you basically want to get rid of those, because when you have a very large file, if you have these extra columns that you're not going to utilize, it's going to basically make it more confusing. The file is going to take longer to load and all this stuff. So basically what we're going to do is we're going to only keep certain columns. And you can do this really easily in Galaxy. And we're going to use their text manipulation tool. So specifically, we're going to click on Cut Columns from a Table, right? And we're going to do the cutting, though, by basically just telling that Cut tool which columns we want to keep, okay? So we're going to, so in the first window, it tells you file to cut up, file to cut, it would be the first data set, this UCSC genome annotation table that we have imported. So we have only one, so it's, the one that's shown up. The operation we're going to run is we're going to tell it which columns to keep. It is a tab delimited file, so we can leave that as is. And we're going to cut by field, so fields are going to be the columns. And then we're going to list a set of columns to cut, okay? Or I'm sorry, to keep. So we're going to keep 2, 3, 5, 6, 9, 13, 14, and 15, okay? So let's see, let me just double check it's the right one. So 2 is the name of the gene. 3 is the name of the chromosome. 5 is the transcriptional start site. 6 is the transcriptional end site. 9 is the exon count. 13 is the full name of the gene, or I should say the official name of the gene. 14 is the coding sequence stat, so basically how well the start of the coding sequence is annotated, or whether it is annotated, and 15 is how well the end is annotated, or if it is annotated at all, if it has a coding sequence. Okay, so those are correct, so I'm going to hit execute, okay? So notice again what happens is a new little tab appears, or rectangle appears, that is basically going to be the output for that function running on that first table, right? And so now, once it turned brown, that job got sent to the Ucsc Browser, and then when it completes, it becomes green. And so you can't view it unless it's green. Now sometimes if the server is really busy, or you maybe have a bad connection, that job isn't gonna run. If that happens, then I would just wait, maybe 30 minutes or an hour and just try rerunning it. Okay, so now let's take a look at the contents of that file. So notice now it looks a lot cleaner, right? We got rid of all those columns that we really didn't need. And now we have only specifically the information that we're looking for and that we're actually going to utilize, okay? Now, again, what we want to know, and the reason we're downloading this file, is to figure out what is the mean and distribution of the gene length in the human genome, sort of the length of genes, right? How long is the average gene? And so in order to do that, we have to do a little bit of data transformation, a little bit of computation, right? So basically what we want is we want to figure out the gene length, again, by subtracting the transcriptional end site, subtracting the transcriptional start site from the transcriptional end site, right? So that would be, in summary, we want to, for every single row, we want to subtract the value in column 3 from the value in column 4, okay? So again, we can go back to our text manipulation tool, click on compute. We're going to add an expression. So this we're subtracting from column 4, column 3, okay? And then we don't want it to round the results. We do want it to skip the header line. So the header line has the name of the field, and so it doesn't make sense to subtract those, right? So we're going to skip the header line. And then we do want to name the column. So we're going to kind of keep the convention of the naming or the format of the names that's in that original NCBI file. So notice that what they do is they don't put any spaces between the words, and they basically capitalize the second word, right? So exon count is merged together. There's no space between those two words, and the C is capitalized. So we're going to give this name, we're going to give this column a name of gene length, okay, with the L capitalized. And then we're going to hit execute, all right? And so again, look how that job got sent here. If we click on it, it just says job is waiting to run. It's gray. Okay, we can basically we just have to wait. Now it's turning light brown because that job got sent to the server. It's getting run. And now a small job like this, it should only take 10 to 15 seconds, right? So if a job like this is taking more than a couple minutes, then there's some issue with the server, or there's a problem with connectivity, right? So you just have to maybe try it in a little later time point. All right, so that data set turned green, right? So that means it's completed. We're going to take a look at the file, make sure that everything got calculated correctly, right? So notice we have all the same columns we had before, right? Except now we have a additional column at the end, column 9. It has the name gene length. And then if we look at the values, just to sort of double check them, they look correct. So let's say, for example, if we look at this gene right here, right, the length of that gene should be, so it starts at, or I should say it ends at position 8,423,832. It starts at 8,352,000, right? So that's roughly, it's about a 70,000 gene, right? So the value that was produced by our formula that we had is 71,429, right? So that makes sense. You should always double check it because it's easy to make a mistake. It's easy to pick the wrong column and not get the right answer. So always, at every single step, you always want to double check that whatever function you run, whether it's in Galaxy, whether it's in any bioinformatics program, or whether it's in RStudio, that it's giving you the answer or the value that you expect it to be, right? It's very important, because otherwise you potentially could be introducing errors and not catching them until it's too late, or until you've already done a bunch of other analysis that you then have to redo. Okay, so now we want to download this as a table, right? And so Galaxy makes it really easy. All we have to do is hit this little file thing called download, right? So we download it, and then you can see down here it's downloading the file. It's a pretty big file. It's a text file. It's a little bit over 10 megabases, right? Now what you want to do now is before you, or I should say, in order to import into RStudio, you want to make sure you don't try to open that file in any other program. Don't look at it. Don't change the extension. Nothing. Just leave it as it is. At most you can basically maybe copy it or move it to the working directory that you're going to be doing your analysis in RStudio if you want to. Okay, but I'm not going to even do that. I'm just going to directly import it from the downloads folder. So now I'm going to go to RStudio. Okay, in RStudio, remember that opening a file and importing a file are two different things, right? Think about it this way. If you want to open a file, you're opening a text editor or some kind of editor, right, just to look at it. If you want to analyze data, you have to import that file as a data set, okay, so that it ends up in a data frame, and RStudio knows to treat those columns as data that it can perform functions on, right? So to do this one, what we're going to do is we're going to go to File, Import Data Set from Text. So we're using the base import function. We're going to pick the downloads folder, right? And then I am going to click on that file that was downloaded by Galaxy, and I'm going to hit open. And so when you hit open, you're going to get a window that has the specific arguments or parameters that you're going to use in that function that's importing that data set, right? And so first thing we're going to do is we're going to change the name. So we basically want to change the name to what we want the name of the data frame to be. So we're just going to do human period genes, okay? Next, what we're going to do is we're going to check all those other arguments to make sure that the import is happening with appropriate parameters, right? So we can leave the encoding as automatic. Heading, we're going to click yes that the file has a heading. So notice that when we had no, it was treating that top row that has the names of the columns as the rest of the data. But if we click yes, all of a sudden it's treating that top row as the name of the columns, which is what you want, right? Because you want those names to refer to the values that are in each column, okay? Let's see, what else do we need to do? We can leave the decimal as, the separator. It's a tab delimited file, right? So one of the nice thing about this this viewer, it's showing you what the input file looks like, right? So you can see that the columns are tab delimited, and then you can see also what it can like a preview of what the data frame is going to look like, right? And so you got to make sure that that it makes sense, basically. So the decimal can be used as default. The quote we can leave as default. Comment we don't have any in there. Missing data and a string. So that's really important. You want to make sure that, however, the missing data is coded in that file you're importing, that you use that code for missing data in this NA strings argument. Okay. Now, I don't think this this table has any missing data, so I'm just going to leave it as default. And then we're also going to click strings as factors. So basically, what that means is that whenever there's a some sort of letter, so in columns that have both letters and numbers, if even one value has a letter, it's potentially going to import that whole column as just characters, so basically just words, right? And if you click strings as characters, instead it's going to import those as factors, so basically categorical data, okay? And so that's important because, for example, one of the ones we're going to be looking at is the name of the genes, right? And so we want to make sure that that column that has the gene names is imported as a factor, and each gene name is a different level, right? Okay, so strings as factors. So now we're going to hit import. And so now it takes a few seconds. And so two things happen, actually three things happen. So first, we had the function that corresponds to the selection in the menu that you made is passed to the console, right? And it's basically sent to R, right? So selecting in the menu import data and clicking the file and then import is basically creating an object human.genes that shows up in the global environment in the data. That's a data frame that includes 166,923 observations of 9 variables, right? So there's basically 166,923 rows in that table, okay? Also what happened is there was a function view that ran, and that basically opened up the data frame viewer, which is this window here, right? And so, you can kind of we can look through it, make sure that everything makes sense. Notice again, each of the columns is named based on that field that was in the original file, right? All the information looks correct. If we look at the structure of our data, name is a factor, chromosome is a factor, transcriptional start site, end site, exon count, those are integers. Name 2 is a factor. Codon start stat and codon end stat is a factor with levels. And and so forth. So if we actually look at to see, okay, how many genes are actually annotated in this, let's see, in this particular file, it looks like it's 38,557, right? Because there's 38,557 levels in the gename file. Now that looks a little low to me, but it appears that that's how many genes are in that annotation file. Okay, so that's basically how you import a table that has a a data set from a track in UCSC and going through Galaxy so that you can actually do some table manipulation, right? Now, like I said in the beginning, we want to look to see what is the mean average length of a gene in a particular or in the human genome. So we can get that really simply. We can do it two ways. So we can run the mean function. So mean parentheses, and then we're going to enter the human.genes data frame, and then we can select the column by hitting the $, and then all the different columns show up, and we're going to pick gene length and hit enter, right? So the mean length of all genes in the human genome is 75,737.1 base pairs. Now, if we wanted to create the distribution, we have to do a little additional step, right? So a distribution in R is referred to as density, right? So we're going to plot. So we're going to do the plot function. Inside the plot function, we're going to include the density function for that same column. So human genes, and then we're going to select gene length, right? And so now we can see the distribution of the human, or so gene length of the human genome is very, very far from normal. I'll put it that way. So there's basically some genes that are quite short, right? So most genes are quite short, and then you have a very small number of genes that are extremely long, right? So the longest gene in the human genome is dystrophin. It's about 2.3 million base pairs long, right? But a majority of the genes are, again, fairly short. They're only, the mean is 70,000 base pairs, and a lot of them are even shorter than that. Okay, so I hope that you found this useful. I hope that this gave you, some information on how you can get data from the UCSC Genome Browser. Remember that everything that in the UCSC Genome Browser, all those tracks are actually data sets that are publicly available for you to analyze, and so therefore you can download them. Going through Galaxy is a great way to go because it allows you to, again, do some basic analysis with the tables, and also, of course, Galaxy has a lot of excellent, the latest bioinformatics tools, so you can actually do a lot of complex analyses in Galaxy as well. And you can very easily download those data sets from Galaxy onto a desktop computer or local machine and get those into RStudio so you can do additional statistical analyses. So again, I hope this was helpful, and I'm going to try to post some additional videos in the near future as well. Thank you very much.